I'll leave one side in case you understand.
So do you want to say anything first? For those of you who have never experienced one of these things before, what's going to happen is Austin is going to talk about his dissertation research. And then all of you will have a chance to ask him whatever questions you want. And after that, we will excuse the hoi polloi, and then his committee, including Drew Lee, and his committee, it'll be his committee and Austin, and we'll grill him some more. So that's the order of events. And I looked on my records today, and this is the 34th time I've done this since I've been at UNC. And I was thinking about my remark yesterday, and I realized that I've always had an easy time doing this job for my students for two reasons. And some of those reasons, one set of those reasons is because there's something common between all my students. And another is there's always something unique. So the common things are, one, I know that they all worked hard, because they couldn't have acquired this much, they wouldn't be here unless they had done 9 experiments that didn't work for every experiment that did. I know, number 2, they thought about everything, because when you're faced with that much failure, you've got to figure out how to fix it. And so all the time, you're doing experiments that didn't work, you fix them, you make them work, and you move on to the next one. So I know they've done a lot of thinking. And because they've done a lot of doing and a lot of thinking, they've all learned something. Okay, so that's what all my students have done. Now, what makes Austin unique? I think, no, this isn't embarrassing. This is not going to be embarrassing. I think what makes Austin unique is Austin is the best NMR jock, nuclear magnetic resonance jock, I've ever had in my group. Not only that, he's a pretty decent mechanic as well. And I think that's important. One reason he can do all this NMR is because he understands how to fix things. And so I always look for an excuse to look at my favorite books. And this is a book called Cannery Road, which is absolutely my favorite book. And it talks about a guy just like Austin. And since I've got the floor here, I'm going to read one paragraph. I should say that Cannery Row is about a bunch of bums who live on the coast of California and work in the cannery sometimes, and they just absolutely know how to live. And at one point, they're going to borrow somebody's car, and they're actually going to go up and get a bunch of frogs, but that's not important for this story. This story is about the car. And it says, probably any one of the boys from the Palace Flophouse could have made the truck run, but they were all competent practical mechanics. But Gay was an inspired mechanic. There is no term comparable to green thumbs to apply to such a mechanic, but there should be. For there are men who can look, listen, tap, make an adjustment, and a machine works. Indeed, there are men near whom a car runs better. And such a one was Gay. His fingers on a timer or a carburetor adjustment screw were gentle and wise and sure. He could fix the delicate electric motors in the laboratory. He could have worked in the canneries all the time had he wished, but in that industry, which complains bitterly when it does not make back its total investment every year, and I should say this is like grants, if you want to see it as an expression, the machinery is much less important than the fiscal statement. Indeed, if you could can sardines with ledgers, the owners would have been very happy. And it was they, as it was, they used decrepit, struggling old horrors of machines that needed the constant attention of a man like Gay. And I think Austin is Gay. And so, it's not my fault.
At least the real name. Thanks, Gary, for calling the game. All right, guys, thanks for coming and listening to me today. So I'm going to be telling you a little bit about a project that I've been working on only for, I guess, a couple of years. This wasn't the mass of the research that I did for Gary. I actually came up with this project. It's very simple. You'll see all the NMR that's in this is very, very simple, but I think it has some really important, I've come up with some really important conclusions from this very simple NMR experiment. So basically what I'll be telling you about today is how protein stability and dynamics are different in a cellular environment, something that has a lot of other proteins and nucleic acids, just a very rich environment. How it's different than just this beaker of water and buffer and salt. So that's what I'm going to be going into and telling you a little bit about today. So to keep in mind for the whole time that I've given this talk, this is my overall conclusion, and the two bolded parts of the important things, that cells destabilize proteins, contrary to predictions. And it does so enthalpically. And I think it does so by interacting with the actual unfolded ensemble of the protein, not the actual folded native state of the protein. So those, keep that in mind as I go through all of these slides. So what we're going to do today is we're going to talk a little bit about protein folding and stability. Just a quick slide on that. We'll go into macromolecular crowding, the old school theory, and then kind of the new thinking for macromolecular crowding. My protein, my model system, I'll show you just a quick slide on it. It's a very simple, small protein domain. We'll go into fluorine NMR briefly, and towards the end we'll get into a little bit more depth on fluorine NMR. And this is actually the molecule that makes all this able to happen easily. This is 5-fluoroindole, and this is my nucleus that you're going to be looking at on all my N-cell and dilute solution NMR spectra. So then we'll get into the actual business of this. We'll talk about crowding the small SH3 domain, and we'll look at some N-cell spectra for the stability and a little bit of dynamics, and then we'll also look at in vitro experiments where we've crowded with polymers and proteins alike to try to understand really how the differences in N-cell stability come about. Okay, so first of all, let's look at folding and stability. So equilibrium protein stability is the fact that proteins exist basically in two states. A two-state folder exists in just the denatured ensemble of states and the one single folded state. So we've all seen the classic folding funnel where we have free energy and then we just have conformational space on the top. So the unfolded state, or what I will designate as U throughout this entire talk, has a lot of conformational freedom. It can exist in a lot of different states at any given moment. But as that protein starts to make native contacts and starts to fold, it falls down this funnel and it hits the one folded native state. So I also kind of redrawn this in terms of just the two states where you have just a jumbled up protein with no real structure, the unfolded state, and then the folded state of, this is actually the SH3 domain, the folded state of the SH3 domain here. And the difference between these two is actually the free energy change, or the delta G of unfolding. So this is actually the energetic difference between the two states. So any two-state folder follows this path. There's no other intermediates that it populates in its folding. It's just the unfolded ensemble and folded state. So we look at this in terms of just a simple equilibria. We have the folded state and the unfolded state, and the equilibrium constant of unfolding dictates how much of each one of those two are in solution at a given time. So if we know this, then we can actually measure and get this free energy voting. So this is the method that we use to get this stability measurement, or this free energy unfolding. So just negative RTL and K, something we've seen since Kim very corrected me, 102. So we've all seen this. Okay, so that's what I'm going to be talking about for a lot of the first part of this talk, is equilibrium protein stability. So we're going to be looking at populations of the folded and the unfolded state of the small protein. So what we're actually interested in knowing is if that difference changes in a cellular environment that's rich with macromolecules. So I already kind of introduced this on the first slide. Traditional biochemistry is studied in just this beaker of water and salt, less than 10 grams per liter of your protein or macromolecule of interest. But that's not exactly what the cell is like. The cell is filled with all types of macromolecules and salts alike, with up to 300 to 400 grams per liter of just macromolecules. And this is actually a little simulation. So this was just a snapshot, but this is a short simulation from the Elcott lab that shows what the cytoplasm actually looks like. This is slowed down 1 million times, and it has the 50 most abundant macromolecules plus GFP as a reporter. So if we watch this movie, you can actually see that these proteins don't have a lot of freedom to move around. They're always near another protein or something else, a nucleic acid of some sort. So they just don't have, it's not the same type of environment as just buffer, where it's just happily floating through solution, not contacting anything. So this environment is expected to change many different properties of proteins. The first couple of years I was here, I studied structure of unfolded segments and disordered proteins in this type of environment. We're not going to go through any of that today. What we're going to focus on today is how stability changes in this kind of environment, and how there could be changes in protein dynamics also in this environment. So first, let's look at the theoretical, what the theory is for this change in stability due to this crowded environment. So traditionally, since the 80s, they've studied this macromolecular crowding by using synthetic polymers like phycol. So what phycol is it's just large branch sucrose polymer. So I have sucrose here, and what you would imagine is for every one of these, each one of these OHs would have another sucrose coming off of it. Very highly branched polymer, thought to be relatively globular and especially inert. And we also have here just this typical equilibrium between the unfolded state and the folded state. So what is thought is if you have a lot of these phycol molecules around your protein, they just take up space. So I've shown here just a Crowder. We're going to represent this as a sphere like we do everything. And our protein of interest here. And what this Crowder does is it takes up space. And there's a certain volume that's excluded to the center of, in this case, the SH3 domain. So I've just shown one Crowder here, but you can imagine if you have 10 Crowders around your protein, then it's not going to have a lot of volume to take up. So what it's going to do is it's going to exist in its smallest form. It doesn't want to be unfolded because that takes up more space than does the native state. So what that actually does is it favors this folded state. So this will be a higher population here, and it will increase the stability of our protein. And I can think the best real-world example of this excluded volume effect is, imagine you're in a hotel on the 10th floor going down in an elevator. You get on the 10th floor, there's nobody else with you, so you can do whatever you like in the elevator. You get to the 8th floor, and 10 people come in. And you basically have to pack together in that elevator. You don't have much area to move in. That is the excluded volume effect. You can't exist in the same space as another person. So that is basically the theoretical idea behind macromolecular crowding. My favorite picture to depict this is these people being packed onto a train. You know they're pretty compact inside that train. So that's one of my favorite pictures. So that was then. And now, in recent years, there's been a lot of research that kind of refutes this. This is purely an entropic effect. There is no enthalpic component to this. And a lot of theory now is starting to say we've oversimplified it. So this slide is all research done in the past in this lab. What we have here is basically a thermometer, the stability of another small protein, common trypsin inhibitor 2, in 100 grams per liter of a various number of Crowders. This is a delta G. So this is the difference between the crowded environment, and buffer. So anything at 0 is the same as buffer. Anything in blue is stabilized relative to buffer. And anything in red is destabilized relative to buffer. So macromolecular crowding was initially devised for phycol polymers, polyvinyl provolone polymers. And what we see is that excluded volume effect is stabilizing our protein. I should also say that the width of this little circle for each of those is the error in the measurement. But more importantly, things that they always exclude volume, but things that have charges or things that can stick to your protein, like bovine serum albumin, another protein, lysozyme, another highly charged protein, and reconstituted E. coli cytoplasm, they all destabilize CI2 relative to dilute solution. So They still have excluded volume, you still can't exist in the same space, but chemical interactions are starting to come up. Recently, this was not in cell, recently in lab there's been in cell stability on the B1 domain of protein G, just another small protein. And what this is actually the delta G of mutation. And then in blue is the buffer delta G for this mutation, and red and green is in cells. And what you see here, the most important thing, is the cellular stability of GB1 is always lower than that of buffer. I think the more important thing here is if you make a charge changing mutation, D to K, buffer, this is a surface mutation, the proteins don't feel each other in buffer, it's pretty much innocuous. There's no real change in stability. But if you put that in cells, there's a real decrease in stability. So that really was the first, I think, experimental evidence that said that these protein surfaces are really important. And that is really where my project's going to build off. My project's going to try to figure out, is this in-cell stability different from entropy or enthalpy? And then also, what's the role of these surface interactions? So this is just in terms of stability, but dynamics have also been probed in these crowded environments. This is from Lewis Kay's group in Toronto, and he's got two projects on this. The first one is a small domain that interconverts between a native state and an intermediate state. And what they can actually do is they can measure this interconversion using some NMR methodology. And it's very interesting. The actual folding path of this protein is changed in lysates. So what I have here, this is a graph of population of the intermediate versus the exchange rate between the two. So between the two conformers. And in buffer, we have an exchange rate of about 2,000 and a population of about 1.5%. But in lysate, our population increases. So the actual intermediate is stabilized. It's more highly populated. And the exchange rate between the two is slowed down. I think that's probably due to just the sticking of the cytoplasm to that. So dynamics changes. They've even used calmodulin. This is just a C13 HSQC. And this is a side chain shift as a function of lysate. They've actually measured binding constants, very, very weak millimolar binding constants of lysate for a protein. So what they've also done, if you throw this into a lysate that has some specific partners for calmodulin, you don't see a spectrum at all. So this really tells the importance of these chemical interactions inside cells. Chemical interactions can change everything. So my project really is trying to figure out entropy or enthalpy. Which one causes this in-cell destabilization? And then which one is it, what's being picked on by the lysate? Is it picking on the folded state, or is it interacting with this unfolded ensemble? So that is where I'm coming in and trying to answer these two questions. So to do that, I came up with this idea while I was helping Gary with some fluorine isotope shifts, helping him make some figures for a grant. I saw this paper that was with the small, only 59 amino acids, a very small domain of a much larger protein. That's been very well characterized by NMR. And in this paper, I saw basically there was one tryptophan in this molecule, and if you labeled that tryptophan with fluorine, you took a fluorine NMR spectrum, you could actually see two peaks. And I immediately thought, well, this is great. One of these peaks is for the folded state. The other peak is for the unfolded ensemble of this protein. And if you can actually measure the population of those two species, you can get a delta G very, very rapid. So the key thing about this protein is that its stability is basically zero. It exists in both states at approximately equal concentrations at the same time in your test tube. So when I saw that, I said, well, we can do that in buffer. Someone's already done it in buffer in the literature, even though it's not a very good looking spectrum. I'll show you some better ones here in a few minutes. I can crowd this with whatever I want. And in 5 minutes, the 5 minutes that it takes to collect this, I can collect, under any kind of condition that I would like to have. So it was a huge time savings, and I thought, well, I can just make a data mountain very, very rapidly with this and hopefully get some important results. So basically, what we have here is we have the folded state and the unfolded state, and the actual area under this peak. So if you cut this out and weighed it, and then cut this out and weighed that, that weight is a relative population, basically, of those two states. All right, so how do we do this? E. coli is actually pretty smart for being something that doesn't think. You actually can just feed E. coli 5-fluoroindole, and it makes this into tryptophan. So basically, it doesn't want to waste its energy synthesizing its own tryptophan. You just feed it 5-fluoroindole, and it's going to turn this into 5-fluorotryptophan, and it's going to incorporate it wherever there's tryptophan that it needs. So it likes to save its energy. And this was one of my first spectra of purified protein that has been labeled by 5-fluoroindole. Looks much better than the one on the last slide. But you see the folded state and the unfolded state. This is in buffer at 25 degrees Celsius. And what we can actually do is we can measure that peak volume, and we can get the population, basically, of that species. And if we can do that, I showed you that on the first slide, the delta G equals negative RT on K of unfolding. Well, the K, this equilibrium constant, is just the population of the unfolded state over the population of the folded state. And we can get that readily from right here. So we can get a delta G in about 5 minutes, maybe 20 minutes tops with this method. If you try to do the same type of measurement with a very stable protein, using hydrogen-deuterium exchange, it can take days, weeks, months to measure one stability. I can crank out a stability in about 5 minutes. So what we're going to do is, instead of just taking one stability, we're actually going to measure these fluorine and MR spectra over a variety of temperatures. Let's take a look at that. Again, we have here just a fluorine and MR 1D spectrum. I've got the folded state labeled and the unfolded state labeled at a variety of temperatures. So you can see at 10 degrees Celsius, there's not much unfolded state populated. As you increase the temperature, the protein starts to unfold, and we actually see some denatured state, and the native state is decreasing as you increase the temperature. What we can actually do is we can plot the free energy of unfolding versus temperature for whatever condition we like. So in black here, I have what it looks like in buffer. And you can't see the error bars in here because the error bars are actually smaller than the data points, and that's from 3 independent preps of this protein that all of my experiments used. So basically, we just average those 3, and then the standard deviation is the error bar that's smaller than the data point. So then what we're going to do is we're going to fit these data points to the integrated form of the Gibbs-Helmholtz equation. And this is where this project really sets itself apart. If you can measure the temperature dependence of stability, then you know all the thermodynamic properties of that protein. You know not only the stability, but you know the enthalpy and the entropy and the delta Cp at these two at any temperature you would like. So you just have to pick a reference temperature. Typically, we pick where the protein melts, so where delta G equals 0 as our reference temperature. And then fit these curves and generate an entropy, a reference temperature, and a delta Cp that defines all the other thermodynamic parameters. I'll tell you a little bit more about, we're going to see lots of entropies and enthalpies, and I'll tell you a little bit more about the error analysis when we actually get to one of those stages. So that's how we do this. In buffer, we see what the stability curve looks like, and I've got 2 other things on here. I have trimethylamine oxide at 50 grams per liter. All these are at pH 7.2 also. TMAO is a traditional protein stabilizer. The protein would rather be near itself than it would be around TMAO. And you see an upshift, basically, of this curve. What that stands for is just stabilization across the entire temperature. Urea is a typical protein denaturant, and you see it's below the buffer curve. So this is just destabilization of this domain in 100 grams per liter urea. So this all checks out. Everyone knows that TMAO stabilizes, check. Urea destabilizes, check. But that's not really what, my talk's not about this in buffer. My talk's about this inside cells. So let's actually look at how we make an in-cell NMR spectrum cap. So I grow these up in M9 minimal media, just a little bit of glucose, a little bit of nitrogen salts, and some 5-fluoroindole. Of course, we want this to actually incorporate 5-fluorotryptophan. What we do is we grow these cells up, we induce expression, and we end up having the cells full of F19 enriched tryptophan. Then what we're going to do is we're going to harvest that cell pellet, and we're going to wash it multiple times. And we're going to resuspend this whole entire cell pellet at whatever concentration we may like. Typically I get 50% cell slurries. And this is what the NMR tube looks like with about 100 mils of culture, all down and resuspended at about 150 microliters of buffer. So this isn't your typical looking NMR tube. Stick it in the NMR, and hopefully we get out a spectrum. This is actually a spectrum of the SH3 domain inside cells, 25. The key to this is we have to take this back out of the magnet, we have to pellet down all the cells, take the supernative, and make sure that there is no signal. So in blue we've got the end cell, and in red we have the supernative, and there's nothing in the supernative. So all of this stuff came from inside the cells. So that's how we do an end cell NMR spectrum. Let's look at this over a temperature range of 10 to 40 degrees Celsius. And there's a few things you notice immediately from this. Probably the most glaring thing that you see is in the supernative, which is in red, you start to see something leaking at around 30 degrees Celsius. The good thing is there's no folded state. There's no folded state that's leaking. So the actual protein doesn't leak. What's leaking is basically a degradation metabolite of this protein. And it's a little bit easier to see if you look at the lysate of this. So this is just each one of these experiments. I've lysed the cells, thrown it back in the NMR. And you can actually see this metabolite peak in all of these spectra. So we actually have to correct for this in our measurements of in-cell stability. So keep that in mind too, that we actually subtract out this peak from the in-cell spectrum to try to come up with a true delta G for inside cells. So the only other question that we have to answer before we can compare this to buffer is what's the pH inside our cells? We all think it's around 7.6, but some of my other one of the other papers that I published has shown that it's really not 7.6. In this case, it's around 7.2. And we really got lucky with this protein because in this inset here, I have the purified protein resuspended at different pHs, 7.5, 6.5, 5.8. And these are all normalized to the denatured state, so all these spectra have the same chemical shift and intensity to the denatured state. And what you see is these peaks get closer together as you lower the pH. So that's basically delta omega here. So we have the chemical shift difference versus pH, and you see we have this curve that I've just fit to a polynomial. The good thing is we can look at the delta omega inside cells, and it's about 450 to 500 hertz, which corresponds, using the standard curve, to about pH 7.2. So the in-cell environment is around pH 7.2 for these cells. The other important thing that we can note from this is, we notice here that pH 7.5 and 6.5 have the same intensity of these two peaks. So the stability is pretty much flat in buffer over that pH range. So if we missed it by 1/10 of a unit or something like that in cells, it shouldn't make that big a difference. Okay, so let's look at what the actual in-cell experiment looks like. So again, we have another one of these curves. We're going to see a lot of these curves for the next few minutes. Free energy of opening, where positive numbers are stabilized and anything below buffer is destabilized, versus temperature. And I have the in-cell spectrum at 25 degrees here, and the buffer spectrum at 25 degrees here. Immediate thing that sticks out to you is it's much broader inside cells. We're going to get into that in detail on the slides. But the most important thing here is cells destabilize the SH3 domain relative to dilute solution by about a half a kcal over physiological temperature range. We didn't go up to 45 just to not hurt the cells too bad in this experiment. So the important thing, this was kind of known from the GB1 work. The important thing here is we can actually split this into entropy and enthalpy by using the integrated form of the chemical solution. So what I have here is all of these data are at 298K. I have the results for buffer and the results for cells. And you can see what the delta G, what that stability difference is, about 500 calories per mole for buffer. And inside cells, it's destabilized. It's almost nothing. The stability is almost nothing. The populations are almost equal. So the important thing here is we can actually split this into enthalpy and entropy. And what we see in buffer is it's 10 kcals per mole for both entropy and enthalpy. And the error bars are relatively large. And I want to tell you why those error bars are a little bit large. This delta G value It has small error bars. Those come from the triplicates. And we're just measuring the relative populations of these two things. It's a precise measurement. To get this delta H, we fit these curves. We generate a delta H at a reference temperature, and we generate the curvature of that line. So we have three parameters with three of their own errors. So we have to also propagate that error. So we have three things that have errors. That's why we have such a large value there. It's not just two populations, it's three uncertainties. Key thing here is, inside cells, we have a decrease in 2 kcal per mole in enthalpy. That is a destabilization term. We also have a decrease in the entropic term, but that's stabilizing. So anytime I have green, that means stabilizing, and red means destabilizing. So what cells do, the inside of cells have this excluded volume. They do have this entropic stabilization, but enthalpy, chemical interactions with the protein in the cellular environment actually cause this destabilization to happen. So I think that's the most important thing in one slide that I've done in graduate school. So I think that's pretty nice when you put that in one slide. But what I'm interested in is trying to understand how this works using synthetic polymers and proteins alike. So we're going to look at more of these curves, but in vitro, not this in vivo, this in-cell NMR activity. So I've just redrawn what you've just seen over here. This is the same thing from the last slide. And what we're going to look at on this slide is how polymers just can't do this same thing. And so this is where all the theory came from, like I mentioned on the first slide. We have sucrose, glucose, and ethylene glycol that we're going to be looking at on this slide. So here we're going to have three more sets of these data come up. Again, all of these are the delta G versus temperature. And we have a lot of different scenarios here. On each one of these panels, the black is buffer and the red is inside cells. So just immediately looking at that, you look at it and you say, well, all these polymers stabilize the protein. There's none of these that are below the black line. That's the knee-jerk reaction of this data, is that polymers can't do this same type of thing as the in cells. But we can really break it down a lot further because we can get this temperature dependence and stability. So in 300 grams per liter of glucose and dextran, just a glucose polymer, and 300 grams per liter of sucrose and its polymer, glycol, we get this stabilization. And what we can actually do is we can find out that this stabilization, by some error analysis and fitting these curves, is actually entropic in nature, just like theory predicts. So glycol, our error bars, can't really let us say much about it. It's not that different from buffer. But dextran, which is in green here, is significantly different than it is in buffer. You get a big stabilization, about 600 calories per mole, and that is due entirely to entropy. There's actually an enthalpic penalty here. So this is that pressure to fold. This is that excluded volume effect coming about. What kind of confused me, still confuses me a little bit, is our results in PEG, polyethylene glycols, at 200 grams per liter. These curves look a little bit fundamentally different than all these others. The sugar polymers, they just shift the curve up, basically. Ethylene glycol shifts it to the left. And what that turns out to be is that actually turns out to be enthalpic stabilization, not entropic stabilization. You can see that here. We have green under the enthalpy, and we have an entropic penalty. So macromolecular crowding, the old theory, it works for sugars, but it doesn't work for all polymers. So really better theories need to be come up with to fit this macromolecular crowding effect in cells and even in what the theory is based on, polymers. So polymers don't really cut it. We can't use polymers to try to replicate the inside of a cell. What we can use, I think, a little bit better is other proteins. So you can buy BSA and lysozyme from Sigma for cheap and resuspend at whatever concentration you want. So again, we have what we've seen for the last couple of slides in cell spectrum over here. This is our free energy of unfolding versus temperature graphs for buffer in black, and then 100 grams per liter crowded experiment. So we just had 100 grams per liter to this experiment, and we see an upshift of our curve. So that's slight stabilization of this protein. Lysozyme, however, is Berry is a very charged protein. It interacts pretty strongly with our protein, which we'll see here in just a few minutes. And it significantly destabilizes this SH3 domain, much like cells do. So I think lysozyme is a pretty good mimic for the insect environment. The problem with using these two proteins is when you do all the error analysis, you can't really slice out the entropy and the enthalpy. There's no difference in entropy and enthalpy compared to the buffer scenario. So we can't say anything besides the lysozyme mimics the cellular environment in terms of the net destabilization. But one of the things I think is very important about this project is it allows us to change the charge state of our protein. So all these proteins have a certain isoelectric point. At any pH above that, they are negatively charged. At any pH below that, they're positively charged. And we can just vary the pH, and we can change their charge. So we have 4 panels here. We have another one of these delta G unfolding, and we have temperature down here for all 4 of these panels. All of these Crowders are 100 grams per liter. The only difference between these panels is the pH. So here's my buffer and cell. I've thrown urea on here too, just as a reference. And this is what you just saw on the last slide. This pH 7.2 experiment crowding by BSA and lysozyme. And the other thing I have on here is the net charge of all of our proteins at this given pH. So the SH3 domain and BSA are both negative. They experience extra repulsion, not just the excluded volume effect, but even more repulsion. So that's where the stabilization arises. And lysozyme is destabilized. It's positively charged. So they actually stick together. And what you see here at pH 7.2, I'm just taking the difference, the delta G at 25, and I'm turning it to this little arrow so we can compare the relative stability differences as a function of pH. So we get about a K-copper mole destabilization for lysozyme pH 7.2. At pH 5.4, we've actually switched the charge state of BSA. So BSA has a PI a little bit higher than 5.4. And so now BSA is positively charged, while the SH3 domain remains negatively charged. And I think this is pretty cool. We see slight destabilization now of the SH3 domain when it's crowded by 100 grams per liter BSA. What we can do when we lower the pH is we can look at lysozyme also, and what we actually see is we see a lessening of the delta G basically. There's not as much charge difference between the two species, and it doesn't stick quite as much. So we see a lessening of that. Then we can go on to pH 3. We can't do BSA here because we think BSA kind of specifically binds the SH3 domain. We start to see more than just 2 fluorine peaks. But what you can see is the SH3 domain and lysozyme are positively charged. And the majority of this destabilization is mitigated. So charge matters a lot. This electrostatic sticking matters a lot in terms of stability. But what you also see is it's still destabilized, even though they have the same net charge. So charge isn't the only thing going on here. I think hydrophobics are going to play a pretty big role in why this doesn't go above the just buffer curve. But that's still research that has to be done. So let's summarize the first part of my talk. Excluded volume always exists. The 2 molecules always take up space. They can't interpenetrate one another. We can't exist in the elevator all packed together. You can't fit 100 people in an elevator, right? But these weak chemical interactions, or this enthalpic effect, dominates inside cells. And I really think that it has to do with this charge-charge interaction between the two. So just brought about a lot of my data together here. We have a delta G thermometer where the width of the circle is the air. And you see cells and lysozyme are pretty similar. So I think lysozyme is one of the best models for the cellular environment. So for the rest of my talk, we're going to be talking about which one of these 2 states matters. This is a unique protein that exists in these 2 states at approximately equal concentrations in solution. So it's one of the few proteins where you can actually measure interactions with both states easily, relatively easily. So for that, we're going to use lysozyme as a model system. Some of these experiments we can't do inside cells. We're going to use lysozyme as a model because I think it's a pretty good model. We're going to use spicol as the polymer model for this, and we're also going to use urea, which is just a classic to nature for the rest of these experiments. So what we're going to be looking at now, we've talked about stability from here on. We're going to talk about dynamics by NMR. NMR is sensitive to dynamics, fluctuations, how proteins move on a large variety of time scales. And we're going to go from really simple to not simple in the last part of this talk. The most easiest and the thing that we're all probably familiar with is line width of our resonances. So if something has a very sharp line, an NMR line, it means it tumbles very fastly. Small molecules, you can even see splitting between adjacent coupled nuclei. It's one of the reasons why it tumbles so rapidly. Proteins have much broader lines, and it actually, if you get a large enough protein, you actually stop seeing the resonance at all because the tumbling is so slow that it just goes into the baseline. The SH3 domain is nice because even inside cells we can see an NMR spectrum. So what we're going to be talking about when we talk about line width is basically the width at half height of these lines. So basically where these two arrows are pointing for the folded and the unfolded state. That's what we're going to be talking about with the most simple is the line width. I'll also use some 2D NMR, fluorine nuclear overhauser effect spectroscopy to measure folding rates. This is a nice protein. It exists in the folded and the unfolded state all at one time, and it's in slow exchange. So we can actually use these 2D experiments to measure the exchange of this folded state to the unfolded state. So that's something I'll introduce to you in just a second. And then we're also going to look at the tumbling time of our protein. We're going to look at T1 and T2 relaxation of our nucleus to get that. Okay, so the first thing I'm gonna talk about is T1 and T2. So these experiments are simple in that they're one-dimensional experiments. We don't have to have a nitrogen. We don't have to use multinuclear NMR for these. We just have a typical two peaks, and we're going to measure the difference in decay of those two. So these two things measure different relaxation properties of our nuclei. I'm going to show you kind of. So basically, when we put something in the magnet, we put a molecule in the magnet. We're going to go into the X, Y, and Z plane. Now, all the nuclei align with the magnetic field. So we're going to look at that as the Z plane. So everything's going to align like that. And we can use radio frequency pulses to manipulate that magnetization. So the first one we're going to look at is T2. So what happens in a T2 experiment is we actually are going to put this magnetization into the XY plane. So now we're just going to have this magnetization against the magnetic field in the XY plane. And what a T2 experiment does is it measures the actual decay of this magnetization in the XY plane. So we basically just evolve time. And we watch this go smaller and smaller and smaller to nothing. So what that looks like is that's going to look something like this. So we have basically a peak intensity versus time. And we can fit this out to get our T2 relaxation, or T2 time. The next one is T1. So this works by measuring the basically decay of this magnetization back to equilibrium along the Z direction. So what we're going to do now is we're going to manipulate this magnetization to go against the magnetic field. Then we're going to evolve time again, and we're going to watch that magnetization decay back to its equilibrium value. So both of these two experiments, this is what this looks like. We just have intensity, and you initially have negative intensity going positive as a function of time. What we're going to do is, looking at these two, is we're going to look at a couple of different emotional properties of this fluorine nucleus using these two experiments. Now, we actually have we get two numbers for each state, a T1 and a T2. And T1 and T2 depend on a lot of different variables, actually. It depends on what we're most interested in, the rotational correlation, how fast that molecule tumbles in solution. It also depends on the distance between whatever is causing your fluorine nucleus to relax. In this case, that's a hydrogen. So it depends on the internuclear distance. So this isn't quite as easy as nitrogen relaxation, where it's dominated by its attached proton. This is a little bit different. It also depends on a couple of parameters that deal with internal motion, basically the time scale and the rigidity of the two bonds. So 4 parameters, we only have 2 numbers. We're going to use some assumptions to basically make these stick. We're not going to vary those. There's some research that has shown that these numbers don't really vary in lysates. It comes from a Lewis case group. So we're gonna say that those two parameters don't change inside cells. And we're gonna fit out just a tumbling time for all of these. So that's what T1 and T2 are. So it's a little bit more complex than line width. But we'll get into some of the data here in just a second. And the last thing that we're going to measure is we're going to measure basically the folding and the unfolding rate of our protein. We're going to do this using a nosy, and most of us probably think of a nosy in terms of through space interaction. That's how we've all learned nosies. But that's not exactly how it works in this case, because a nosy can also measure chemical exchange. So this pulse sequence basically has a variable exchange time where we can watch the folded state turn into the unfolded state. So here's kind of how this works in a little, I think, less detail than that. So this is just our one-dimensional experiment. We have this T1 here. That allows us to take that into 2 dimensions, which allows us to turn that into a 2-dimensional plot. And you can imagine here, this peak is the folded peak in 2 dimensions. They're on the same chemical shift, 48 and 48. And the unfolded is here. 49 to 49. So these are just the self peaks. What this experiment allows us to do is change the exchange time. And what you notice here, as you increase the exchange time, you start to see a peak not on this diagonal here. It comes off the diagonal. And what that really corresponds to is that it corresponds to magnetization that was initially in the folded state, exchanging onto this unfolded state. So what we can do is we can fit these four peaks to a series of equations. So we just have the area here of all the peaks versus time. We can fit this to a series of equations and get a folding rate and an unfolding rate. So this protein is really nice because it lets us figure out tumbling times and folding rates in cells and cell-like environments. So the first thing we're going to look at is the line width. So this is all at 298 K, and these protein crowders are 100 grams per liter, and phycol is 300 grams per liter. And we see I've also labeled the folded and the unfolded state here. And you've already seen what cells look like. You see a lot of broadening. But I've not shown you what lysozyme VSA and phycol look like. You see a lot of broadening with lysozyme, and you see that destabilization, that denatured peak, that unfolded peak is much more intense than this folded. But what we can look at this is we can actually look at a little table of these values and kind of understand this a little bit better. The line width in just buffer is around 30 hertz. It's pretty narrow line. And the first thing I want to draw your attention to is what phycol does. So you don't see a whole lot of broadening with phycol, and I think a lot of that broadening really is due to, phycol is right here, due to an increase in viscosity. So phycol, 300 grams per liter, is about 25 times as viscous as water. And that's going to slow protein tumbling down, and that's going to broaden your line out. So you see some broadening, but it's just not nearly as much as it is in, say, lysozyme or in cells where the viscosity is not that much more than water. So lysozyme's viscosity is basically the same as water, 1.3 times. But both of the states broaden out significantly, not because of viscosity, because of these weak chemical interactions. VSA doesn't really do much. It's relatively inert at this concentration. And inside cells, viscosity is 2 times that of water, but we have an order of magnitude change in our line. So in cells, it's not due to viscosity, it's due to this steam. And what we can actually do, I think, is measure this in a little bit more detail using tumbling times and folding and unfolding rates. So what we have here is we have the tumbling time in nanoseconds per radian. So that means the higher the number, actually the slower it's tumbling. It's taking it more time to turn a radian. We have this for the folded state and the unfolded state. So let's just start at the bottom. Buffer and urea both have tumbling times of around 3 to 5 nanoseconds per radian. So they tumble very quickly. Phycol is this viscous solution. It tumbles just a little bit more slowly than it does in buffer, but really not that much. I think the key here really, sorry about that, it's due to viscosity. The key here really is the lysozyme and cell data tumbling times. And you can see that it slows tumbling of both states, both of those things do, lysozyme and in cells. But you notice that both of them, they slow down the tumbling of the unfolded state more so than it does the folded state. Cells is much more so. I should have also mentioned in the beginning that the width of that circle is the uncertainty of that measurement. So We don't have really good measurements inside cells for this. We do for lysozyme. But this is key because this says that these chemical interactions dominate the unfolded state. So the unfolded state is basically just open. It's not well folded. It's not, all of its surface area isn't buried. It's just able to interact with whatever proteins are around it. I think it shows this even better if we look at the folding and unfolding rate constants in these conditions. So what we have here is we have our rate constants for folding and for unfolding. In buffer, we're about a folding rate of about 3 per second and about 1 per second unfolding rate. It's a stable protein. Folding is faster than unfolding. Let's look at urea first. Traditional denaturant, it destabilizes proteins by slowing folding, and it also speeds up unfolding. So it kind of kicks both ways. It doesn't want your protein to fold at all. This is 100 grams per liter. Phycol slows both folding and unfolding. And there have been other groups that have shown this also. This is partly due to viscosity. I think this is also partly due to the basically the excluded line effect action. So you can think of this as a protein. It's got a lot of surface area in the unfolded state. It can't fold as well because the viscosity is a little bit higher. And then once it's folded, it just wants to stay that way. I think the more important thing here is the lysozyme data. We see a relatively large error bar for the unfolding rate of lysozyme, but it doesn't change compared to buffer. They're within error line. What does change is this folding rate. The folding rate drops up by almost an order of magnitude. And what that tells me is, again, it's about surface area. The unfolded state has a lot of surface area that lysozyme can just stick to. It's electrostatic in nature, is what I showed you on a few slides ago. So basically, once lysozyme is stuck to the SH3 domain, the SH3 domain can't fold. It's just kind of stuck along the outside, and it can't actually fold into its form until it kind of dissociates with the protein. Okay, so that's my second big thing. So let's put all this together and have a few conclusions. Cells destabilize proteins enthalpically. We've seen this. I've shown you this 4 or 5 times now. You're all probably really tired of seeing this. But cells do this enthalpically. We turn to model systems, basically, to try to understand this. And what we found is that polymers can't replicate the cellular interior. So I've got these arrows, these equilibrium arrows, are actually scaled properly for the folding and unfolding rates. So this is what it looks like in the loose solution. We have a stable protein; it wants to fold. Polymers, they slow down folding because of viscosity, but they really slow down the unfolding due to this compaction effect. But this just can't match with the in-cell situation. What I think really can, and what can tell us a lot about this enthalpment effect, is my results for lysozyme. What proteins do is proteins have weak interactions with the unfolded ensemble because of its surface area. And basically they just stick and they won't let the protein fold. And that's how they actually destabilize the protein, because they populate this, they favor this a little bit more. All right, so I want to thank Gary, first of all, for letting me be in his lab, always listening to my ideas, letting me kind of do things, maybe are bad ideas, but he lets me do them anyway. I want to thank Greg for always being around, helping me with NMR. Greg really helped me with some of the NMR experiments I did before the scoring stuff. So he really helped me out with full sequence on that. Drew, I want to thank you for talking to me relaxation. Drew's lab was the first lab I was in here at UNC, so I got to learn protein NMR through Drew and Paul. So thank you guys for that. Max, who couldn't be here on my committee, but he's talked to me about this project recently and has helped me out thinking through some of that. Linda's helped me out with pH stuff. I've had a very supportive committee, actually. I've talked to all of you all at some point. Linda helped me out with some pH stuff on other papers, and I've talked to Nancy since day one in Gary's lab. We were on the same floor, so we've talked about stuff throughout my time here. The work for this project was done. A lot of it was done with me and my undergrad sitting in the lab arguing with each other over what we should and shouldn't do. You mainly heard me yell at Larry all the time. Annalise helped me out with some of this relaxation stuff, so thank you, Annalise. And Michael was a German exchange student. This guy right here, he took this project and went his own way with it recently with reverse micelles, and other things that I don't really know much about. So it's been really nice working on this. I want to thank everybody else also. People that can't be here, Will and Mahana, really helped me come up with this project, and also helped me with a lot of stress relief throughout the whole time, and all the rest of you guys. It's been fun also. So thank you. All right.
Questions? First, I just want to make a comment. I think this is another triumph for gay America.
2 weeks in a row.
Yeah, this is great. So everything we've worked on has been these very small proteins. Can you speculate about the applicability of what you've discovered as you increase the size of the protein?
Right, so I think size will definitely scale, and these larger proteins, in terms of surface area, are going to have a lot more surface area in the unfolded state for other things to work with. So I think that's going to be one of the big things. I think it will. I think the effect might even be more so for larger proteins, just because they have so much more area to work with.
So in that case, why do proteins fold at all themselves?
Well, I think it's pretty delicate balance. I think proteins basically were evolved in a certain way to make them be active, to be able to fold and be active. One surface mutation, like I didn't do any surface mutations with my stuff, but like with Will's stuff, one charge mutation changing from a negative to a positive changed the stability by a lot. So I think that proteins are really tuned specifically to be able to have do their job, but also to be able to be recycled easily, basically. That's my thought on that.
So I'm going to interject something here. Let's say that Austin used a 33 kilodalton protein, which is the average molecular weight of protein in E. coli, and he found one that had a stability of 0 kcals per mole in buffer. These experiments, I bet you dollars to donuts, would absolutely not work because that protein would just never fold. It would get stuck to everything else in the cell. And so one reason this works, this whole project worked, is it was a small protein with free energy near zero. And this is why, getting back into the big picture of biology, this is why if you make destabilizing mutations in proteins, they're often associated with disease because they irreversibly unfold and then aggregate.
Larger proteins have lots of, sometimes intermediates too, that would make this basically impossible. Anybody else?
So your delta G versus T plots are like less curved in cells?
Sometimes they are less curved in cells, yes. It is a little bit less curved. So that, if you can see the 95% confidence interval, if I showed you the error of actually the curvature, delta CP, they're within the error buffer itself. So this is just on the best fit line. They do have similar delta CPs, but the in-cell delta CP has much larger error involved in it.
Okay.
So yeah, that's one of the things that isn't perfect.
You could go crazy with the idea that they were different.
Right, but I think you could also talk, that also has to do with surface area, right? So, but I think there'd be a lot of other experiments you have to do that to kind of back that up. Josh?
Do you halt protein synthesis before taking these in?
Yeah. So we had chloranthanol within our washes, and I said I'll wash 3 times. All those have chloranthanol in them.
Because I was curious about the heat shock response when you're measuring from 30 to 40 degrees Celsius. There could be a dramatic change in stability.
With Gary and I have talked about that also. That's one reason why we don't want to go higher than that, because that could be part of the reason why it's not a perfect curvature. Also, there could be some kind of basically heat shock response going on there.
What's the concentration of your protein compared to any heat shock proteins?
Well, the concentration of our protein is millimolar for sure, so it's way higher. But that's also part of the reason why we see more of this leakage of this metabolite at higher temperatures. Cells are being under stress.
What's the concentration of SH3 in cells? You might have mentioned it.
Yeah, it's a few millimolar, say around 2 to 3 millimolar.
So you haven't really tuned the different concentrations of that in cells to see.
No, we haven't done any experiments where we can, control the amount. So each one of these cells probably does have a little bit different amount of protein in it also. So that's another thing that, I don't know, I guess a pitfall. But no, we haven't done any experiments to, try to control the amount of expression. Typically, it's always around 2 to 3 millimolar.
But do you measure it every time?
So the best way that the way that I look at that the best is just by comparing the overall intensity of those two peaks. And it's normally very constant, actually, the way that I do it. If somebody else went to the lab and did it, they might get something different than I get. But typically, whenever I do this experiment, the overall integrals are all the same, or close to one another. So there's not wild differences between the experiments.
Okay, I guess we're down to just the committee. Let's thank Alston.